Greetings everyone, and welcome to today's Gen webinar, which is entitled Rapid QC Assessment of AAV Viral Vectors with Stunner. Today's webinar was made possible through sponsorship from Unchained Labs. As always, I'll be your host for today's event. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which has been at the vanguard of the life sciences industry for the last 40 years, bringing you the most updated information on the newest tools and technologies. Now, it's almost impossible to overstate the value of adeno-associated virus, or AAV, as an indispensable tool for gene editing and gene therapy endeavors. Yet, for investigators to use this tool effectively, they need to be able to characterize the critical quality attributes of AAV rapidly and in a high throughput manner. Now, combining advanced analytical techniques like UV-Vis spectroscopy, static light scattering, and dynamic light scattering allows researchers to evaluate the attributes of their AAV samples quickly and using very minimal amounts of precious sample. And in today's presentation, we're going to hear about some case study examples which harnessed a unique device from Unchained Labs called Stunner that combines the previously mentioned analytical techniques. But first, let's meet our presenters for today's event, who are going to provide us with all of that detailed information. Now, first, I'm going to turn the presentation over to Dr. Kevin Lance, who is the Director of Analytics Marketing at Unchained Labs. Now, Kevin has an extensive background in bioengineering with expertise in biophysical characterization of proteins, nucleic acids, viral vectors, and LNPs. Kevin will provide a brief introduction before he hands things over to his co-presenter, Dr. Audrey Chang, who is the Executive Director of RightSource at Charles River Laboratories, where she works with a team to provide biomanufacturing testing services at the manufacturer site for biological therapeutics, including vaccines, blood products, monoclonal antibodies, recombinant proteins, gene therapy products, and cellular therapeutics. Now, Audrey has an accomplished scientific career spanning more than 25 years in both government and industry settings. And today, Audrey will tell us how her and her colleagues use the Stunner platform to examine multiple AAV samples from at-scale manufacturing campaigns at Charles River Laboratories. And then Audrey will pass the presentation back to Kevin, where he will provide some more technical insights into how Stunner works and how it might be useful for your AAV projects. But before we get the webinar underway, I want to remind the audience that we'd love to hear from you in our Q&A session. The Q&A session is a lot more fun and exciting if you send in questions. Now, you can do this at any time during the presentation. You don't have to wait until the end. So if something pops in your head and you have a question for Audrey or Kevin, just type it in and hit submit. We'll try to get to as many of your questions as possible. All right, with that said, I'm going to now turn things over to Kevin to get things rolling.
Thank you for that introduction. And thanks for attending our talk on rapid QC assessment of AAV viral vectors with Stunner. Today, Audrey and I will both be discussing different elements of AAV QC. Audrey will be introducing adeno-associated virus and giving a taste for the current state of QC testing and characterization, and go into the current unmet needs for analytical tools in a manufacturing environment. Then I'll pick up the ball and introduce Stunner, a tool that addresses several of those unmet needs, and go into a few case studies examining the details of a recently published paper in Vigene Biosciences and walk through some additional data that's only seen here. And now I'll hand it off to Audrey.
Thank you, Kevin. So I like starting talks with this slide, which shows the progress of the type of therapies now available to us. In the 60s, we started with the simple chemically defined small molecules, and from there we moved to recombinant proteins, followed by the larger monoclonal antibodies, and to today's exciting cell and gene therapies. More recently, nucleic acid drug products such as the mRNA COVID vaccine, which we all know about today, and gene editing modalities present a bright, promising future. But with each new modality, the manufacturing of these products presents challenges with respect to evolving manufacturing platforms, new raw materials, and new understanding of therapeutic mechanism of actions. And this is where the data from analytics and assays will provide the added insight. There are several viral vectors, including lentivirus, adenovirus, but AAVs, adeno-associated viruses, is the most popular one to date for in vivo applications. This is due to AAV's lack of pathogenicity and its ability to persist in vivo. And AAVs are also quite versatile. Many vectors are being custom designed to target specific tissues. The table I'm showing here is taken from last year's FDA Advisory Committee meetings briefing document, and it says that in a 5-year period from 2015 to 2020, 24% of the gene therapy studies worldwide involved AAV. In that same 5-year period, the FDA saw 99 INDs using AAV, as represented by the blue bars, and an even greater indication of what's to come is the increase in the number of pre-IND AAV-related meetings, as shown by the orange bar. And of course, we all know the 2 AAV products on the market, with more to come. But regardless of the modality, all drug products intended for humans must be manufactured under current good manufacturing practices, including quality control testing as governed by regulatory agencies worldwide. And although differences may be present, they all call out the same testing principles. 1. The test performed assures product acceptability. 2. The data obtained from the test must fall within preset, scientifically based specifications. And 3, QC testing is part of an overall control strategy for the manufacturing program. And these tests fall under basic categories to provide answers each time you manufacture it. Identity, is it the right product? Purity, what manufactured and processed byproducts are present? And these can be host cell related impurities such as genomic DNAs or process impurities from reagents used in the manufacturing process. Potency, how much do I have and what is its activity? And equally important, does the measured activity data correlate to the relevant biological activity? Safety, is the product free of unwanted bacteria, mycoplasma, viruses? And appearance and general quality attributes that include product-specific characteristics ranging from something as simple as pH to more of the complex secondary structure. And it is often these assays that are used in stability studies. But looking at QC testings for AAV, this slide shows where AAV biology comes into play. AAV biology dictates that during manufacturing, the target product, which is the vector containing the therapeutic gene of interest, will be produced, as shown in the picture full. But also present will be particles that have no genome package, called empty. And the percent empty can vary from 50 to 90% and is dependent on the manufacturing process. Additionally, there is also the potential that partial sequence or other variants that might package host cell DNA can be produced. So why is this a concern? Most obviously is that an empty particle is unable to deliver the therapeutic benefit, but from a safety perspective, there is a concern that the heightened immune response from a patient's total capsid exposure is from all of these particles. Additionally, the potential of illegitimate packaging of host cell genes can be concerning if they package antibiotic resistant gene or oncogene that will be unintentionally delivered to the patient. Therefore, it's expected that this critical quality attribute, this CQA, that defines the percent ratio between full and empty particles in a batch is established. And that specification, that percent number, should be based on your risk assessment and data generated from product development runs, engineering, and manufacturing runs. And this expectation to characterize the amount of empty capsid as a percent ratio of full to empty particle is clearly outlined in the recently released FDA guidance on gene therapy. Now, if you read this FDA document in hopes that you will get what that percent threshold number is, you will be disappointed. There is no number called out. I made an earlier reference to an FDA advisory meeting that was held in September last year. And this was a meeting that was dedicated to understanding the toxicology risk from an AAV vector. And this percent full empty ratio was the most prevalent CQA that was discussed. And this is very timely. It's timely to have this meeting because with the clinical successes we are seeing with AAV and the rapidly growing number of clinical investigations we are seeing, AAV therapies will undoubtedly move from a rare disease indications to larger common disease indications. So it is important to have these discussions and data now. And how do we get this data? These are some nice assays that are used to look at empty full particles. There are others, but these are the common ones. If you put the assay parameters side by side, as in this nice 2021 paper by Gimpel, one can see each assay has its nuances. Let's start with analytical ultracentrifugation, AUC for short. And it is the current gold standard method for determining the percent full. Basically, this is a specialized ultracentrifugation run with an optical, I'll call it an eye, that can monitor and collect a series of sedimentation velocity profiles over time during the run. And from this, you can identify the expected peaks into full, empty, and also partial peaks. And to date, this is the best method for partial. But it is not a high throughput method and does require a specialized instrument. The next one, transmission electron microscopy, TEM, is a direct visualization method, and it gives a picture that clearly distinguishes what a full particle looks like from an empty one. But like AUC, TEM is not considered high throughput. It does require a specialized instrument and is dependent on several very critical sample preparation steps to get a nice image. The last two methods, ELISA and PCR, are methods you will see more commonly used in a standard lab. So the ELISA will detect the capsid protein, which is present on both the empty and full populations, and the PCR will only detect particles that have the genome and can be used to represent the full population. And although these methods are more accessible, the data is less than perfect because it represents more of a broad stroke view. But all of these methods take time to produce the results that range in hours, and that doesn't even include the sample preparation time, which make it less than ideal to be called a rapid QC method. Why is a rapid QC method? And a lot has been said about the need for rapid quality control, and it's being driven by these new modalities that have a short shelf life. But in essence, time is valuable to all. Holding a batch, waiting for QC results before moving downstream is undesired, as is moving forward at risk. So therefore, all manufacturers stand to benefit from getting reliable QC data quickly. And this is fueling the concept of real-time release testing, RTRT, getting the data fast. If you recall my first slide where we started with a simple small molecule, real-time release testing is a reality for them today. And there's a lot of interest in making RTRT a reality for biologics, which will rely on advancements in analytics in the quest for this perfect QC assay. So when asked, what would you wish for in this perfect QC assay? Well, this perfect QC assay would be performed as fast as possible, using as little volume of my precious material as possible. I should be able to run many samples in a batch, and the results that I obtain will be 100% reliable no matter what time I perform it. Additionally, the perfect QC assay will work with all sample types and produce data to the sensitivity and specificity that I require. It should be easy to perform. It should produce a complex data, ideally on several attributes. Shouldn't cost much, Shouldn't have to worry about supply chain issues or backlog. And finally, this assay, and I'm doing air quotes here, should be regulatory accepted. And I want to make a note here that assays are never regulatory accepted. It is a product that is accepted. And so we could probably debate on the placement of the red X's and blue check marks, but suffice it to say, no such assay exists today. But we're in a good place for three reasons. One, regulatory agencies are open to innovation. All of the new guidance documents that are coming out refer to implementing new methods. But two, data is needed to implement new methods. And so you remember the foundation of quality control is based on science. And number three, there is a lot of exciting innovation happening in our industry today. And by collaborating and working together, sharing knowledges and best practices, we can progress our industry. So to tell you more about our collaboration, I'm going to turn it over to Kevin.
Thanks. Let me start by first focusing in on two technologies, DLS and UV-Vis. Each of them tells a part of the story for AAV characterization. DLS gets you info on capsid titer and aggregation, but can't say anything about what's happening with DNA. On the other hand, UV-Vis gets you the total amounts of protein, DNA, and can link those to empty fill ratio, but doesn't give you any info about if any of this is contained in capsids. Each gets you valuable info, but neither tells you everything. So what if you could combine them to get a whole story? Those two technologies are what Stunner brings together to calculate answers on capsid titer, empty flow ratio, and aggregation with only 2 microliters of sample in less than a minute. And it does this using no labels, reagents, or standards. You just use the sample you've got. So let's dive into this and how this all happens. Getting data from such a small sample volume starts with Stunner's 96-well consumable plate. In the plate, each well is actually a microfluidic circuit that lets you use that 2 microliters to get information from two technologies, UV-Vis for concentration, shown on the top, and DLS for sizing, shown on the bottom. SLS intensity data is also read during a DLS experiment and gets you valuable insight, in this case on titer. The plate can be read from, or can be used to read from 1 to 96 samples at a time. And in our case, Stunner's AAV quant application is what we'll use looking at the data with Audrey to combine all the pieces of information into the full story about an AAV. Let's first drill down into how this is all possible from 2 microliters. Here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, we've already added fluorescein, and as you can see, to load another sample, you simply add in 2 microliters into that input well, and the sample wicks into the plate. Here's what those microfluidic circuits look like before and after being read on Stunner. On the left, you can see those loaded samples, and on the right, you can see the samples after being measured with Stunner. The 2 diamond shapes are the microcuvettes, where 1 sample is read for UV-Vis, SLS, and DLS. What this also shows is, after pumping, all the sample remains in our consumable, so there's no contact and no contamination between the sample and the actual pump system. Here's what the raw DLS and UV-Vis data look like for an AAV sample on Stunner. Checking out your AAV with DLS tells you what shape your sample is in, if there are only capsids or if aggregates have started to ruin things. DLS also lets you figure out how much SLS intensity is coming from just your AAV capsids. You can measure how much area under the curve is coming from the green peak on the left, while the gray peak is aggregates and larger stuff. What we want is the percentage of green area versus the total area under the curve. UV-Vis, on the right side, brings information on how much total protein is in your sample and how much DNA is in there. Based on some simple info you tell Stunner's software, it uses the balance of protein and DNA to calculate the empty full ratio and percent full. What Stunner is doing is shown step by step here. First, on the left, DLS identifies the capsids, the aggregates, and how intense the signal is from each of those populations. So let's say the area under the curve that's green is 70% of the total area. In the middle, that info then helps you split up your SLS signal from a total value into the amount just attributable to capsids. So this is analogous to taking a total brightness and splitting it into how much of that brightness level from light scattering is coming just from capsids. And finally, UV-Vis brings in info about empty-full ratio, plus protein and DNA totals to provide the last piece of the puzzle. These data combine in a few steps to get to an accurate capsid titer answer. The SLS data by itself can actually be used to figure out capsid titer to a rough approximation, but for any given SLS intensity, you can figure out only a capsid titer range. The puzzle isn't complete yet because full capsids scatter more light than empty capsids. So if you have full capsids, that means you actually have fewer capsids present, and vice versa. If you have empty capsids, your capsid titer will be higher. So what we need to do is bring in the empty-full ratio from UV-Vis to let us pinpoint exactly where the capsid titer really is. That's how DLS, SLS, and UV-Vis combine to give fast, accurate answers on capsid titer. Here's the whole story of a lot of AAV data Stunner gathers if we told it in one graph. Starting from the left side blue bar, which is the protein in the sample, UV-Vis got you the total amount of protein, and here we're expressing it as how many possible capsids all that protein could make. By adding in DLS and SLS info, Stunner splits up that information into the light blue bar, which is aggregates and extra protein junk, and the dark blue bar, which is that total titer for the AAV capsids that we got from the previous slide. On the right-hand side, in the green bar, the process follows the same steps for DNA. The total height of the green bar is the total amount of DNA expressed as the total number of genomes that you could make, the viral genomes. Light green is extra DNA, and dark green is the number of full AAV capsids. The proportion of dark green, which is full capsids, and dark blue, which is total capsids, is the percent full value for the sample. Here's that same data showing the exact values Stunner delivers to help you know your AAV inside out. We can see the total capsid titer and full capsid titer shown at that bottom table, and the excess DNA and protein present is also expressed in terms of equivalent capsids or equivalent vector genomes that amount of protein and DNA could be used to make. Now we'll pivot to the following case study that comes from the work that Audrey and I were a part of along with the other co-authors on our teams as a collaboration between Unchained Labs and Vigene, which is now part of Charles River Labs. The publication explores the use of Stunner's AAV quant application as a rapid QC tool for AAV. When an AAV product is pure and stable, the composition dictates its specific optical behavior. For example, given absorption of light and given scatter, and the combination of all these products is kind of an optical signature unique to that AAV product that you made that can be used to monitor for consistency in the performance of GMP manufacturing runs. Especially more resource-intensive assays are being performed. So if you get the answers that you've historically gotten across these different optical measures, then you know that you're producing the same product. As shown in the last few slides, Stunner measures the optical behavior of AAV to also meet several of the unmet needs Audrey discussed earlier, namely that characteristic UV-Vis absorbance, absence of absorbance from impurities, second, the expected size and size distribution by DLS, and finally, the titer and empty-full information that's synthesized from these data. In this next example, also from the paper, we have taken a refined AAV sample whose total UV-Vis absorbance is shown on the left. Right away, we can start digesting this for clues to show that this isn't yet fully purified. Classically, that might be the ratio of our A260 versus A280, and we can compare that versus a historically expected value for that ratio to try to understand what the balance of protein and DNA is in the sample. But thankfully, Stunner has a better way to do that. It can use unmixed deconvolution algorithms to break down the total spectrum, shown on the left, into the individual spectra that add up to that total signal. So in other words, the black line on the left is the same as the black line on the right, but with Stunner's unmixed algorithms, we can break it down into what's shown in the green line, which is the DNA present in this AAV sample, the blue line, which is protein, and purple, which is all impurities, but here it's composed mostly of iodixanol. Because Stunner can calculate the absorbance of each of these components, it can quantify them too. You'll see those values in the table on the right at the bottom with equivalent vector genomes per mil for DNA, equivalent capsids per mil for protein, and a volumetric percentage for iodixanol concentration. If we instead had used Na260 to estimate the amount of DNA present without accounting for the other components present, the result actually would have had approximately a 60% error in this case, something that Stunner and its unmixed tools help you avoid. To demonstrate the linearity of Stunner, a twofold dilution series was used for an AAV9 vector starting from a 1E14 Vg per mil sample. These samples, shown here, were tested in triplicate. The results indicate high linearity of the full and total capsid titer results, with R-squared values to 0.999 and above. Precision is shown by error bars that often aren't simply visible, as low as 0.5% CVs and maxing out at 11.8% CV for the sample on the left. Percent CVs for the empty AAV sample on the right are between 1.4 and 4.4%, often much better than you'll find with a comparable ELISA kind of assay. Results for total capsid titer and full capsid titer for Stunner's AAV quant application are compared here against ELISA and qPCR respectively. Titers were determined for 6 different AAV constructs of 4 different serotypes, and you can see across this graph that the full capsid sample titers from Stunner were within 30% of the qPCR results for most samples. On this graph, the second sample shown here is actually an empty control that showed an approximately 3% signal by qPCR and a DNA signal by Stunner that was at or below the lower limit of detection, agreeing with its intention of being used as an empty control sample. Both qPCR and Stunner's full titers were highly reproducible, with qPCR percent CVs below 5 and Stunner percent CVs below 6 for full samples. And Stunner's total capsid titer was also typically within 30% of the ELISA titer. Reproducibility for Stunner's total titers was similar to ELISA titers, with CVs at 4% and below for Stunner and below 6% for ELISA. While not fully explored in this paper, differences between these assays can also be used to learn more about an AAV sample in complementary and orthogonal ways. For example, PCR methods are sequence-specific, but UV-Vis methods are able to measure the total amount of DNA present in a sample. And each of those two tools can be useful when you're looking for specifically the amount of viral genomes present or for how much total DNA is packaged in your sample of AAV. In the next case study exploring some data outside of the Gen Biotech publication, I want to look at the questions Stunner can answer at different steps in the AAV manufacturing process. Because Stunner requires a purified sample above a concentration of 1E12 capsids per mil, the samples we'll be looking at come from the downstream purification steps in the AAV production process. When testing a filtered sample from viral harvest all the way through a final product, getting answers on purity, titer, empty full ratio, and aggregates is critical at every step of the way. Because of the low volume requirements and the speed that Stunner delivers answers, it can help confirm if things are within normal operating parameters or if there's a problem going on with any of these questions. If we take a sample from just after each step of the process, this is what we see. In filtered viral harvest, the signal from DNA and protein is high, and that's a very clear measurement shown by the first blue and green bars on the bottom left of the slide. Stunner indicates most of the sample is likely not part of intact AAV virions, since the dark blue and green bars are low, but there's so much free protein and DNA here that purification is really needed before conclusions are drawn. There are a later step in this process, ultrafiltration diafiltration, or UFDF or UFDF, then removes a lot of that high protein and DNA load, which is likely non-viral impurities. That's shown by the decrease in the total height of the blue and green bars at the UFDF step. Subsequent clarification and filtration steps further remove these non-viral components and aggregates. You can also see the final filtered sample concentration has increased since the full capsid and total capsid titers indicated by the dark green and blue bars have increased. If we look at the empty-full ratio for the last two steps, you can see it stays nearly the same. That's the gold and purple bars on the right. Those last two steps are consistently the same with full and empty samples. This confirms that the last purification steps are indeed just removing free protein and DNA and concentrating the product, not removing full or empty capsids and changing the balance. So in this case, the final percent full value is 32%, and there is a full capsid titer at the end of the process of 2.5E13 Vg per mil. All of the data shown here was performed in triplicate on 3 different samples for each step of this process. So it's 9 different reads, which the stunner measured in about 20 minutes, allowing for much faster insights into a process than running a qPCR or ELISA assay. Since stunner is also gathering DLS data on these same samples, we can watch the average size change and do a qualitative check to find out the answers to a few questions on size and aggregation. If we want to know when aggregates are being cleaned up, DLS shows how the sample progresses step by step until it reaches a nicely monodispersed single peak. Or alternatively, DLS can also tell us when the sample is cleaned up enough that we can start relying on other analytics that are sensitive to aggregation. Okay, that concludes our journey through 2 real-world case studies using Stunner for rapid AAV QC. It shows that Stunner helps meet unmet needs with fast, low volume, high throughput, easy, comprehensive, and cost-effective results, where Stunner needs purity but conserves time and sample when assaying AAV titer, empty-full ratio, size, and aggregation in development and manufacturing environments. It's an assay that's applicable to multiple serotypes, genome constructs, and purification strategies, and presents data that's complementary and orthogonal to other current techniques such as qPCR, ELISA, TEM, and AC. And because it's fast to generate, that means any kind of assay looking at Stunner will be able to track and trend data over time, and especially how it compares to these other techniques, and build the correlations and expected values for any process. Lastly, future study on this is needed on the advantages and disadvantages of each method. So for example, when qPCR was needed to troubleshoot a problem versus when a full capsid titer on Stunner is needed to give you a quick answer or troubleshoot a problem. And the optimal approaches that combine any given sample and purification strategy with the appropriate analytical strategies. With that, I want to thank you for your kind attention, and Audrey and I will be happy to take any questions.
Audrey, Kevin, wonderful presentations that really highlight the usefulness of Stunner. So really appreciate that. Massive amounts of data too. Really appreciate you guys sharing that with us. To the audience, the Q&A is coming up in just a few moments. So if you have a question for Audrey or Kevin, don't hesitate to send it on in now. Just type your question into the Ask a Question box and then hit submit. Okay, we have a bunch of questions that come in already for Audrey and Kevin, so let's try to get through as many of them as possible and start the Q&A session. Bear with us for just a moment as we make that transition into the Q&A.
All right, everyone. Thanks for joining us for the Q&A session. We have a bunch of questions for Kevin and Audrey, so let's get to it and try to get through as many of these as we can. First question we have is for Kevin. Kevin, an audience member would like to know, how does the Stunner method compare to other DLS methods?
Okay, so the short answer is that the Stunner method will be more precise than other DLS methods. And the reason for that is because we're using UV-Vis as part of its calculations, especially to figure out, the empty-full balance of capsids present in the titer. That's going to be a very precise and a very accurate method, since there's a wide range of ODs that, different full or empty capsids could have. Other DLS methods rely on refractive index to calculate their titers, and empty capsids and full capsids only have a very narrow dynamic range from across refractive index. So UV-Vis just gives you a much more granularity and resolution when you're looking at samples. That allows you to be more accurate and precise with your titer.
All righty, thank you for that. Next question we have, I think, is going to be for both of you guys. So the first part of the question the audience member would like to know, can Stunner detect partials? And then the second part says, do the speakers feel that partials will become necessary? Maybe Kevin, we can start with you.
So what Stunner does with partials is it will kind of see them as contributing slightly more to full or slightly more to empty. So it doesn't have a third bucket that it says is partials. It just says one bucket for full capsid and one bucket for empty capsids. And then if you're looking at partials, then what you're probably going to want to see is change in your expected results over time, but for, say a standard that you do have completely full AAV with.
Yeah, so I'll take the second part. What was do we feel that it would become necessary, the partial characterization? Correct. Okay. So it is the current method, the only method right now that can detect partials or reliably is going to be your analytical ultracentrifugation. But this kind of goes back to my slide, the ultimate QC assay. You kind of want something that could detect all. And we just don't have all of the analytical tools right now. But it does. I do think that, looking at partials and just understanding the biology of AAV will become more important as we learn more and more about the impact of manufacturing. So it's sort of a chicken and the egg. We have a method right now. Is it something comparable that can do something quickly? Perhaps not, but it will be important, I think, to at least understand what partials, whether it be the partial AAV vector or even the partials, including unintended packaging of genomic DNA.
All right, thank you, Audrey. Kevin, back to you. Another question. An audience member would like to know, what sample prep do I need to do?
Okay, because Stunner's using DLS and light scattering methods and UV-Vis, there's no sample prep that you need to do for downstream methods that are already purified. If you just have your AAV ready to go in your vial, then that's all that Stunner needs to do to see it. I can see there's a couple other in-process samples questions coming up, so we'll tackle those later.
All right, thank you, Kevin. Audrey, back to you. This is an interesting question. An audience member would like to know, what do you think about a consultant company's recent call for the FDA to set a % threshold of full empty in guidance documents?
So I think that's an interesting question. Yes, so there has been a call to say, well, put out a number. Just put out a number that the industry could go by. And I understand the call for that to try to move the industry forward. I think a better call would be, let's open up about what % we have based on our manufacturing process, based on our indication, and make that more available so that we can really do this more data-driven, more risk assessment, and to come up with that number rather than just sort of arbitrarily drawing a number and having everybody now work to that.
All righty, thank you, Audrey. Kevin, back to you. An audience member says, I'm working with a custom AAV and not a default serotype. Will that still work?
Yes. So in Stunner software, we put a lot of effort into making it very, very easy to just sit down with any kind of custom AAV that somebody has. So if you have a naturally occurring serotype, you can pick that from a drop-down menu. But if you need to go in and customize the amino acid sequence, the VP ratio, the post-translational modifications present, or the length of your payload of your genome, then all of these are very easy to change in Stunner software, and things that you could have one serotype AAV5 for a couple wells, and the next few wells can be your own custom serotype, and the next wells can be back to AAV9. So that's really easy to do on Stunner.
All right, and another question for you, Kevin. What limitations does Stunner have? Is it possible to measure in-process samples?
Okay, so for Stunner, what you're going to want to do is have a sample that's above the titer of about 1E12 Vg per mil. So that usually puts you into somewhere in the downstream processing environment. That's one requirement. The other requirement is that you want to, for the best results, you want to have a sample that's been somewhat purified. So if it has a lot of free DNA floating around in it, it's easy just to treat that with a nuclease and get rid of it, and that's going to give you the best results. It can still deal with those samples, but you're going to want to have a conversation with us about how to, analyze that properly. The ability to measure in-process samples. Oh, sorry. Yeah, Audrey, I'll do the short answer here. You can expand on it. Basically, as long as you hit that minimum titer, those samples just have to be cleaned up. So, similar to extraction, like before a PCR test.
No, I like the concept of doing in process, because what you're trying to do is really characterize your process, right? And you want to be able to get data on that quickly. So similar to a PCR, where you do an extraction and then you get your PCR titer, you could do a sample cleanup like Kevin is saying, and then get your stunner number. And what's nice about this is that, whether it's exactly the right number for empty full, it's more of a handle on understanding your process, that you're getting a consistent number with this method through your process. And additionally, if you're going to do changes, you can use this instrument to show if there's an impact on that number. So I know the instrument, what we're talking about here is, how can we use it for percent full empty, but the bigger question is, how can we use this to show that we have control, that we have consistency in our manufacturing steps from step to step. So that's a value as well.
All righty, thank you for that, guys. Next question we have, Audrey, actually, it's going to be back to you, so stay with us here. And let's would like to know, is the analysis of AAV aggregates from monomers a critical quality attribute? And if so, how do you reliably measure that?
So aggregates I would consider it a critical quality attributes for not only AAV, but actually for all biologics. And there's reasons for that. The idea that you have a higher risk of immunogenicity, or maybe you have different conformational changes, and perhaps even different secondary structures that present a different profile. But again, there's lots of different methods that can determine aggregates, such as the one we're doing here. And there's some that are very sophisticated that really give a clear picture, and there's some that are give you an idea of what you have, as we have shown here with Stunner. So the idea is that you have a lot of tools. Pick the one that you want to do with it, and then stick with it, and so that you're able to correlate the data.
All righty, thank you for that. Next question is back to Kevin. Kevin, bear with me for a minute. This is a little bit of a long question, but an audience member says, how accurate is sizing with DLS compared to direct imaging with TEM? AAV sizes determined with TEM tend to trend on the low end, while sizes determined by DLS trend on the high end, over 70 nanometers even. Could this be due to averaging of bulk samples and not a single particles? And can you comment on how accurate DLS sizing is for small particles like AAV?
Sure. So DLS is very accurate for sizing these kinds of particles. And when you compare sizes versus TEM, what we see is pretty much what the question asker says, which is TEM will give you a directly measured size of about 25 nanometers, and AAV will be somewhere between 25 and 30 nanometers in diameter. And so a lot of that difference is going to be just related to the different behaviors that are being measured. One is looking at diffusion through a liquid, and the other one is looking at measuring size. And I would say that the preference is to do this with DLS because you're measuring AAV in solution then, and you're not going through any kind of drying or dyeing process that you have to with TEM samples, which can definitely impact your vectors and disturb any aggregates you have or other things like that. As far as seeing larger sizes, I've seen AAV samples across the spectra from better, PDI, like more monodispersed than analytical standards, and I've seen AAV samples that have tons of aggregation. So it really will depend on how well behavior sampled is, but DLS will give you some pretty good insight into that.
All right, thanks for that. Kevin, another question for you. Audience member would like to know, does the system have 21 CFRP11 software?
Sure, yeah, so for working in a GMP environment, you need 21 CFR Part 11 capabilities, and yes, Stunner has a software add-on that makes that really nice and easy and keeps the software very much a pleasure to use.
All right, and we got another one here for you, Kevin. Audience member would like to know, or Perminder actually asks, says, can we use cesium chloride or inoxidinol purified samples with Stunner?
Iodixinol. So you can use cesium chloride. There's not much of a problem there when you're doing that sort of purification for AAVs. For iodixinol, Parminder's right, it absorbs a lot of UV light. So what Stunner can do kind of depends on how much iodixinol you have present. At higher concentrations of iodixinol, Stunner will quantify your iodixinol for you so you can keep track of how much is there or how much you buffer exchanged away. And then it will still use its light scattering methods to give you a tighter range that your samples could be. As your iodixinol concentration drops lower, then Stunner has some pretty nice unmixed deconvolution algorithms in there that can separate out the signal from the iodixinol from the signal from the protein and DNA of AAV capsids, and then still apply that in combination with light scattering methods to get you to the same titer and empty full information that you would get with a sample not containing iodixinol.
Next question we have, an audience member says, compared to AUC and qPCR, what typical variability exists? Audrey, did we lose Audrey?
Here I am.
Oh, there you go.
Here I am. Yeah, you would think we all know how to do this after COVID here. But I put a reference in one of my slides that there was a nice paper recently that kind of did a side-by-side comparison of the variability that you would expect. And qPCR, the variability is different. And that's mainly because, you're doing it to a standard, right? You're referring it to a standard. But maybe I can expand on this answer, and the way that you compare assays and methodologies, either ones that currently exist or ones in the future, is really going to be through what I call well-characterized reference materials or well-characterized standards. And until we have those standards that we can use and basically show the behavior of these standards across the different methods, that's going to be really valuable data. And then rather looking at the different ones and variability. So that would be my answer. There's a nice paper out there, but we can do more by doing some comparability studies with reference standards.
All righty, thank you for that, Audrey. Kevin, back to you. Another question. How do you differentiate between in-solution and in-capsid cargo as it affects the amount of light scattered by the capsids to make further calculations?
Okay, so that's why this sort of wrinkle is why having both detection technologies and Stunner is so valuable, because UV-Vis will be able to look at, first the balance of empty full capsids or percent full just by how much protein and DNA is there, but it'll also be able to look at the total amounts of protein and DNA that are present. So when you start looking at the total amounts of protein and DNA present by UV-Vis, and the total amounts of capsids detected by light scattering, you can compare those two and use them as a check against each other to understand if you're measuring something that makes sense. Say, for example, you can't measure more capsids than you can make with the protein and DNA that's there, right? Like if you have only, X amount of protein, you can't make 5X capsids out of that. So that's why having both these technologies is really, really valuable. Because if you have something that's, say DNA free floating in your media, then that will show up by UV-Vis, but by light scattering methods, that will scatter light much less intensely than AAV capsids will. So those two technologies just pair really nicely with each other in these methods.
All right, and it looks like we have time for one last question here from Leo. Pretty timely question considering the state of the world at the moment. And I'd like to know, how does this method work with non-viral particles such as RNA LNPs?
Okay, so when you get to larger size particles like LNPs, you no longer can use light scattering to determine titer, but you can still use DLS to look at size on Stunner, and you can still use UV-Vis to look at how much RNA is loaded because those same deconvolution algorithms that we're using to separate out protein and DNA for AAV, you can use to separate out RNA signal from turbidity and then from anything like cholesterol that also absorbs light in your typical LNP. So you don't get to quite the same level of also getting titer answers or empty full answers, but you still get very nice low volume, high throughput size and quantification of the total amount of RNA present.
All right, thank you very much, Kevin and Audrey. Appreciate that. With that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues. We always recommend that. I'd like to thank Kevin and Audrey again for their great presentation, very informative. And I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Unchained Labs for sponsoring this webinar. Hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy.